Welcome to Lecture 9 on the topic of animal genetics. This is part of the Future Farming Technologies subject, which is a component of the Bachelor of Agriculture and Technology that is offered at both North Melbourne Institute of TAFE and Melbourne Polytechnic. Please visit our website at www.nmit.edu.au for further information on this course and other courses that we offer. My name is Dr. Nikki Cooley. In this lecture today, we are going to introduce animal breeding. We're going to look at some considerations for animal breeding. We're going to touch on the differences between population and quantitative genetics and how they relate to animal breeding. We will look at molecular genetics used in animal breeding, artificial insemination and embryo transfer techniques, and economics and breeding. We will be looking at some of these concepts through case studies or actual industry commercial examples. This lecture goes towards our understanding of learning objective number 4 and learning objective number 5 in this subject. Put simply, animal genetics is a technology used to improve animal breeding. Traditional breeding is not a new technology, but modern breeding and some of the associated technologies is. The goal of animal breeding is to select the best animals for breeding. The best animals are the animals which are genetically superior to the others with respect to the traits that you are considering for selection. There is a term called breeding value which is used extensively in animal breeding. This term relates to how much an animal is worth as a breeding animal. It is a very good guide when you are looking at determining what parent you should use in a cross. I'd like you to pause the video now and watch the following YouTube video on an article that was filmed about a breeder that bought a dog at a lot of money and why he chose to do that. There are a number of key decisions that is important in animal breeding. The following two slides summarizes some of these concerns. On this slide there are 9 components which confront breeders when considering their breeding options. These are: number 1, to breed or buy replacement stock. That is the first decision that you should consider. Number 2, which breeding enterprise do you pursue? For example, are you interested in straightforward breeding? And or are you interested in the production of crossbred sires and dams, for example? Number 3, what is the improvement? This may seem like a simple question, but in reality it is actually complex. What is it that you need to do in order to maximize profit for your breeding objective and programs? Once you have identified these, some of the other decisions will become more simple. 4, what recording system will you use? It is important in breeding programs that you keep comprehensive notes. That is because breeding by its nature is one of complex. There are many decisions, many questions, and many outcomes that you need to record and keep a track of. What to cull? In order to optimize your breeding program, you will have to cull some of your outcomes. What to select? To maximize genetic gains in progeny, grand progeny, etc. So what do you mate? Including the age at the first mating, mating structure and use of artificial biological technologies all have to be considered. How effective is the whole breeding program? You have to continuously review, maintain cash flows and maximize long-term return on investment while keeping a close eye on what your business is doing from a day-to-day perspective. And how do you merchandise your product? This is both the seed stock and the commercial product you need to consider. These 9 components come from Animal Breeding, the Modern Approach in 1992, edited by K. Hammond. The following are some notes on the key decision in animal breeding. You may wish to examine the handout notes, as the outcomes of these are clearer on that than they are on the screen. Take some time to read these and review them before continuing the lecture. There are two principal types of genetics, that is, of population genetics and quantitative genetics. Population genetics is the study of the behaviour of genetic variation in populations. This focuses on gene frequencies such as mutation, mitigation, selection and random drifter frequencies, while quantitative genetics comprises the other major component of population genetics and the optional area of animal-plant breeding science. The vast amount of these traits of interest are quantitative, that is, polygenetic. It is both of these components that are important for animal breeding, as both will determine the outcomes of your breeding program. Molecular genetics is the study and the use and manipulation of genetic differences at the molecular level. It is important in animal breeding as it is a very powerful technique, and it includes much of the new technologies. Therefore, we can conclude that in order to optimize animal breeding, we will incorporate quantitative genetics, simple descriptive Mendelian type genetics, and molecular biologies, technologies which will inform all of these. There are some simple concepts in animal breeding that genetic differences between breeds, crosses, and between animals within breeds and crosses for the productivity and product quality traits. In order to explore these simple concepts, we are going to look at a suite of case studies. The first we shall look at is on sheep breeding. Please read the following article on high-tech New Zealand breeding years ahead. Please pause the lecture before continuing. Artificial insemination is regarded as a revolutionary technology, as it has significantly aided dairy farmers and their ability to breed. There are stories of how early artificial insemination significantly boosted cow reproductive qualities, particularly in the milk production side. Initially, semen could be collected and stored for just 4 days, but by the 1950s, due to the ability and experimentation with liquid nitrogen, it was found that semen could be frozen for an indefinite period. This has revolutionized this technology. We will be exploring this technology in further lectures. The development of embryo transfer technique is another milestone event in cattle breeding. With the arrival of this technique, it has now been possible to pass the genetic traits of top quality female animals to future generations. Embryo transfer technique can be abbreviated to ETT, and it involves the removal of a fertilized egg from the reproductive tract of a special breed cow and placed into the reproductive tract of a second cow. This ensures that the top quality characteristics of both a superior quality bull and a superior quality cow are inherited by the offspring. Advancement in this technology has been made possible due to freezing and storage of fertilized eggs. A research on an 8-day-old fertilized egg that was split to produce 4 genetically identical calves has bolstered the belief that the same day it will be possible to impregnate countless number of cows by splitting fertilized eggs from a superior breed female cattle. Cattle breeding will reach a new level of efficiency if this indeed becomes a reality. The second case study that we are going to explore is part of an FAO series of documentations which looks at the challenging global technological context for breeding of poultry and pork production. The following link can lead you to this resource. There will be a tutorial on this link, and therefore once you complete the tutorial, I would like you to insert your notes here. On Moodle you will find another article on the advances in cattle breeding that have been summed up by CSIRO. I'd like you to pause the lecture and take some time to read this documentation. It won't take you long, it's not good. It does, however, provide a really good review of where the technologies are currently. Please take some notes from this article and insert your findings into your lecture notes here. I'd like to present a further case study about the program Breed Plan. This is a breeding program which is aimed at enhancing Australian beef cattle breeding. It is an international beef cattle performance recording and evaluation scheme which is based in New South Wales. It has been operating since the mid-1980s and it is regarded as National's beef recording scheme. It is a modern genetic evaluation system for estimating the true breeding values of cattle and estimated breeding values. Both of these are important parameters for any good breeding program. The link on the screen is a short overview of Breed Plan. Again, please pause the lecture, have a look at this link and add to your notes here. There is no doubt when you're looking at breeding technologies, you certainly should be looking at the economics of your breeding technologies. It is essential to understand both the long-term and the short-term gains. Also understanding what your commodity gains are and where the market is currently going and where you predict it to be headed are all important components to factor into a breeding program. The fundamentals for profitable production are listed below: the market to be supplied, the biological diversity, the productivity which equals the output per unit input, and product quality. The components of the economics of breeding are covered in other subjects, so we will just touch on them in a concept here. The following diagram is a visual representation of the genetic differences of two traits for either a meat or dairy breeding program. The illustration shows you how the inputs are linked from the trait genetic differences that can be measured all the way through to the phenotypic performance characteristics. You may also use this diagram as a summary of the lecture and the components that you need to consider in this lecture. You may want to pause this recording and take a moment to study. So in summary, you should now be able to describe what animal genetics is. You should be able to use a number of different case studies to show recent technological advances and the differences and inputs of each new technologies. You should be able to understand that economics and breeding is important. On your lecture notes, but not presented on the slide on the screen, is a list of resources. These include mate soil optimization, mating allocation, genetics of reproduction for the female influence, genetics for reproduction for the male influence, the relationship between body composition and calving rate, effect of genetic differences in fat net and feed intake on efficiency of weaner production, the beef CRC DNA test for growth, feed efficiency, carcass and reproduction performance, and finally the Australian DNA test for the POLD gene. These resources are all YouTube links to videos that summarise and give you information on these topics. This is not part of the subject per se, but if animal breeding is an area that you hope to get into, I highly recommend you spend some time exploring these different resources, as they will be very useful in highlighting some of the most advanced components of the technology. This brings us to the end of this lecture.